This set of notes is about enzymes. Enzymes are a type of protein. I like to call them helper proteins because they help reactions happen. Just some quick vocabulary before you get started. First, enzyme. Enzyme is a type of protein. Please remember that it is made up of the macromolecule protein. Another word is substrate. This is what the enzyme does work on. So substrate is what the enzyme does work to. For example, there is an enzyme down here. The yellow piece that fits into the enzyme is the substrate. Next are products. This is what the enzyme makes after the reaction. For example, the products of you making a pizza would be a pizza. That is what's made in the end. Lastly is active site. That's where the substrate fits into. You can see here the active site on our enzyme example down here. Again, enzymes are a type of protein. And their function or their job is to speed up chemical reactions in the body. Most of these reactions would not be able to happen without an enzyme. Some examples of those chemical reactions are building molecules. This is called synthesis when you build the molecule. So for example, if we have this yellow atom and this red atom and we're putting them together, with the help of an enzyme, we have put these two to be one complete product. Another example is the breaking down of molecules. We call this digestion. This is what happens when you break down food that you eat. So now it starts as a molecule, and with the help of an enzyme, it's broken down into two separate atoms. These are still a product, even though they're not together. Here's an animation of what an enzyme and a substrate interacting looks like. So the substrate fits into the active site. The green piece is being digested or broken down into two parts. Enzymes basically make all chemical reactions inside your body happen, whether it's building molecules or breaking down molecules. These two types of reactions would not be able to happen without an enzyme. So if you think about it, you wouldn't be able to digest or break down your food without those enzymes. We call enzymes a catalyst. A catalyst is something that makes it happen faster. This here is a graph showing the reaction of the enzyme and how much energy it takes. The black line is before there's an enzyme. So before the enzyme here, let's say it takes 100 units of energy. But with the help of an enzyme, when the enzyme's able to lower the energy needed, let's say the reaction can happen with only 50 units of energy. So each protein has a specific shape in order to fit a certain substrate. So each enzyme has to be the right shape in order to do the job. Think of it as like a lock and a key. I can't use my key to get into someone else's house. Enzymes are named for the reaction they help. So for example, sucrase breaks down sucrose. Protease breaks down proteins. Lipase breaks down lipids. And DNA polymerase builds DNA. These first three are examples of a digestive reaction. whereas the building of the DNA is an example of a synthesis reaction with enzymes. All of the enzymes end in ASE. So if you're reading something and you're not sure if it's an enzyme or a substrate, if it ends in ASE, it's an enzyme. For example, protease is an enzyme that breaks down proteins. When protease levels are low, it causes the acidity in your body to be lower. And this is what causes osteoarthritis, or the breaking down of bone tissue. So as you can see here, there's a normal knee with a lot of tissue and a lot of cushion compared to an arthritic knee, which looks very irritated. And you can see that the tissue here is really broken down. In the reaction, enzymes aren't used up. They have a special shape so that they can be used again and again. So those enzymes are reused for the same reaction to do the same job every single time. For example, looking at this picture, we have a yellow molecule, that's the substrate. The purple Pac-Man looking thing is the enzyme. The active site is where it goes into the enzyme. And the products are what is made in the reaction. So when we talk about enzymes, we say the lock and key model. Earlier I said if I used my key to try to get into someone's house, it wouldn't work because keys have a special shape. The shape of the protein is very important for how that protein or how that enzyme will work. There is a special enzyme for every reaction in your body. Some things that affect enzymes are temperature. There is an optimum temperature. Optimum means best or favorite. This means that the enzyme is able to bounce around as much as possible and touch as many substrates as possible. So, for example, human enzymes like to operate within this range of 37 degrees Celsius, which is the temperature of the human body. If the temperature is raised too high, this denatures the protein. When a protein is denatured, it loses its shape. Think about it as like if you have a piece of pasta and before you boil it, it's a long, straight stick, but after you boil pasta, it turns into a loose, noodly shape. Lower temperatures can cause the molecules or the enzymes to move slower, so then they're not bumping into as many substrates. For example, here's a graph comparing higher temperature to the reaction rate of the enzyme. So looking at this graph here, you can see that as temperature is getting hotter, that the reaction rate is going faster. So the higher the temperature, the faster the reaction. Right here, where the peak is the highest, this is the optimum temperature. This is where it's the best. Optimum is best. So this is the enzyme's favorite temperature to operate at. And then as it gets too hot, the reaction slows down, which denatures the protein or changes its shape. Another example is pH. It's the measure of how acidic something is. It can also denature the protein. Most human enzymes are between the pH of 6 and 8 because this is the pH of blood in most places in the body. Another example, though, is pepsin, which is an enzyme in your stomach, which likes the pH of 3 because your stomach is very acidic. Here's another graph. So looking at this graph, as pH is increasing or getting more basic, the reaction rate is changing. So for example, the optimum temperature, optimum pH of this enzyme is about 7. Here's comparing stomach pepsin, which likes a pH of the maximum of 3. So try to see if you can think of what the pH or the optimum pH for pepsin is. In this graph, it looks like it's about 2 0.5. And then there's trypsin, which is inside of your intestines. What is the optimum pH for trypsin? It looks like it's about 8. For enzymes, what matters the most is shape. Shape is what matters for the enzyme to do its job. Think of this as a lock and a key or a puzzle.